Hello dear friends, hearty welcome once again. Dear students, in this our lecture we are going to study a very little topic, that is mutations and genetic code. The title of our today's topic is Mutations and Genetic Code. Now, what is genetic code? We all are aware, because in the same chapter initially we have discussed in very detail. Genetic code is nothing but it is the basic genetic information present on DNA for the process of protein synthesis or synthesis of polypeptide in code language and in the form of nucleotide sequence. So we are aware about the meaning of genetic code. Now, what is mutation? Mutation म्हणजे काय? Mutation is defined as it is any sudden, irreversible and inheritable change in the structure of DNA. That is, whenever it occurs, any sudden change, inheritable and irreversible change in the nucleotide sequence of DNA, that is structure of DNA, is known as mutation. Now we know that the basic information for the protein synthesis is present on DNA, and that information is in the form of particular sequence of nucleotide. So obviously, whenever it occurs any mutation, whenever it occurs any sudden change in the structure of DNA or in the nucleotide sequence of DNA, it subsequently affects the process of protein synthesis. It subsequently affects the nature and type of protein synthesized by the process of protein synthesis. मी आता असं म्हणत होतो की जेनेटिक कोड म्हणजे काय हे आपल्या सगळ्यांना माहिती आहे, आणि जेनेटिक कोड म्हणजेच प्रोटीन सिंथेसिस साठी आवश्यक असणारी जी माहिती आहे, इन्फॉर्मेशन आहे की जी डीएनए वर कोड लँग्वेज मध्ये असते, आणि ही माहिती मेसेंजर आरएनए वर ट्रान्सक्राइब केली जाते. मेसेंजर आरएनए ती माहिती रायबोसोम जवळ घेऊन जातो आणि रायबोसोम त्या माहितीनुसार प्रोटीन हा तयार करत असतो. पण ज्या वेळेला म्युटेशन्स घडून येतात, म्हणजे डीएनएच्या रचनेमध्ये काही सडन चेंजेस घडून येतात, त्यावेळेला पर्यायाने प्रोटीन सिंथेसिसवर परिणाम होतो. कारण डीएनए वर झालेले चेंजेस, डीएनएच्या रचनेत झालेले चेंजेस, दे आर ॲज इट इज reflected in messenger RNA and subsequently in the protein. Let me explain you. Suppose here is a double-stranded DNA molecule. Here I am going to show that DNA molecule as a straight ladder with two anti-parallel strands. And as we know that DNA consists of numerous nitrogen based pairs. Now I said that whenever it occurs any sudden change in the structure of DNA, that is mutation, it leads to the alteration in the structure, nature and type of protein. First of all, let me tell you the different types of mutations which affect the protein synthesis. Different types of mutations which can alter, which can modify the genetic code and also affect the process of protein synthesis. सुरुवातीला या ठिकाणी मी तुम्हाला म्युटेशन्सचे वेगवेगळे प्रकार सांगतोय की ज्यामुळे जेनेटिक कोड मध्ये बदल होतो. जेनेटिक कोड मध्ये बदल होतो याचा अर्थ काय, तर डीएनए वर असणारी प्रोटीन सिंथेसिस साठीची जी कोड लँग्वेज आहे तिच्यामध्ये काहीतरी बदल होतो आणि पर्यायाने प्रोटीन सिंथेसिस वरही परिणाम होतो. There are three major types of mutations, such as the first mutation is called as gene mutation or may be known as point mutation. The addition, deletion or replacement of only single base pair in the gene is known as gene mutation or point mutation. Please look at the diagram. Consider here, this is supposed to be one gene. A particular segment of DNA which carries information for RNA synthesis. Consider these are the different nitrogen base pairs A, G, C, T, T, A, G, C, T, whatever it may be. I am going to show here complementary nitrogen base to the A, T, C, G, A, T, C, G, A in this manner. Now in this gene, if it occurs either addition or deletion or replacement of only single nitrogen base pair, then it is called gene mutation or point mutation. Means according to this diagram, if only this single nitrogen base pair if get deleted, if get lost, or another nitrogen base pair, suppose G, C, get added in this gene, or this base pair is replaced by any another base pair, it leads to the little change, little modification in the structure of gene, and hence it is known as the gene mutation or point mutation. Here point stands for mutation or alteration in the structure of gene at very small level. So it affects the genetic code and subsequently protein synthesis. How is that? We shall study after some time. Second type of mutation, addition or deletion of DNA chunks at the time of crossing over. Addition or deletion of DNA chunks, chunks in the sense pieces, at the time of crossing over. We know that crossing over takes place at the time of meiosis, and during crossing over it occurs mutual exchange of segments between the non-sister chromatids of homologous chromosome. So whenever it occurs the crossing over, we know the segment of DNA of one chromosome is exchanged with the another homologous chromosome or its homolog. Ultimately it leads to the change in the structure of DNA. So it is also a sort of mutation, and obviously it can change, it can alter the genetic code. Crossing over घडून येत असताना होमोलॉगस क्रोमोझोम्सच्या नॉन सिस्टर क्रोमॅटिड्स मध्ये त्यांच्या म्युच्युअल सेगमेंट्सची देवाणघेवाण होते. पर्यायाने त्या ठिकाणी जे डीएनए चं ओरिजिनल स्ट्रक्चर आहे त्याच्यामध्ये बदल होईल. पर्यायाने जेनेटिक कोड मध्ये बदल होईल and subsequently affects the protein synthesis. And third is jumping genes. First of all, let me tell you briefly the meaning of this term, jumping gene. We have already studied that the major part of DNA of human genome is of non-coding type. Major part of the DNA of human genome is in the form of non-coding DNA. Non-coding DNA means it does not possess any information for the process of protein synthesis. Such DNA is known as non-coding DNA, may be called as the non-functional DNA, may be called as the extra DNA, or it is supposed to be the non-useful. Means, suppose here is the DNA, consider again the same DNA, consider here is a DNA. Now in this DNA, suppose this much part is coding DNA because it possesses genetic information for the process of protein synthesis. But the next part is non-coding. Non-coding means it does not possess any information for the process of protein synthesis, and such non-coding DNA shows number of repeated sequences of nitrogen bases, shows number of repeatedly occurring sequences of nitrogen bases. आणि या नॉन कोडिंग डीएनए वर रिपीटेडली ऑकरिंग नायट्रोजन बेसेसच्या सिक्वेन्सेस आढळतात. म्हणजे समजा या ठिकाणी आपण म्हणूया की हा जो नॉन कोडिंग डीएनए आहे तो मी इथे बाजूला घेतलेला आहे. कन्सिडर हियर इज सपोज टू बी ए टी टी ए सी जी सी जी जी सी टी ए. Now here I have mentioned one sequence of nucleotide pairs or one sequence of nitrogen base pairs, that is AT, TA, CG, CG, GC, TA. Now the same nucleotide sequence occur repeatedly, maybe few time or maybe many time. याचा अर्थ विशिष्ट प्रकारची न्यूक्लियोटाइड सिक्वेन्स ही पुन्हा पुन्हा त्या ठिकाणी रिपीटेडली त्या नॉन कोडिंग डीएनए वर आढळत असते. अँड इट इज प्रेझेंट ॲट डिफरंट साइट इन डिफरंट इंडिव्हिज्युअल्स ऑफ द सेम स्पेशीज. आणि अशा प्रकारच्या या ज्या न्यूक्लियोटाइड सिक्वेन्सेस आहेत, नॉन कोडिंग डीएनए वर असणाऱ्या या ज्या न्यूक्लियोटाइड सिक्वेन्सेस आहेत ज्या रिपीटेडली प्रेझेंट असतात नॉन कोडिंग डीएनए वर, त्या मात्र एकाच स्पेशीजच्या इंडिव्हिज्युअल्सच्या जिनोम मध्ये वेगवेगळ्या ठिकाणी आढळू शकतात. म्हणजे काय, सपोज दिस नॉन कोडिंग डीएनए हॅव्हिंग अ पर्टिक्युलर रिपीटेडली ऑकरिंग नायट्रोजन बेस सिक्वेन्सेस इज प्रेझेंट इन माय जिनोम ऑन द 10th क्रोमोझोम. कल्पना करा की या ठिकाणी मी आकृतीमध्ये दाखवलेले नायट्रोजन बेस सिक्वेन्सेसचा हा जो भाग आहे, नॉन कोडिंग डीएनए जो आहे तो माझ्यामध्ये 10व्या क्रोमोझोम वर आहे. बट इन अनदर इंडिव्हिज्युअल मे बी इन यू द सेम नॉन कोडिंग डीएनए with same repeatedly occurring nitrogen based sequences present on some another chromosome, maybe on 20th chromosome. Means there is a variation, there is a change in their position in the genome of different individuals of the same species, and as they show difference, variation in the position in the genome of different individuals of same species, that's why they are called as a mobile gene or may be called as the jumping gene. Please don't get confused. They actually does not jump from one place to another place, but these jumping genes they can copy itself. ते स्वतःचीच कॉपी तयार करतात. They can copy itself, and that gene formed after copy is incorporated at another place in the genome of same cell. आणि त्या जंपिंग जीनची जी नवीन कॉपी तयार झाली आहे ती मात्र त्याच पेशीच्या त्याच सेलच्या न्यूक्लियस मध्ये असणाऱ्या जिनोम मध्ये दुसरी कुठेतरी जाऊन इनकॉर्पोरेट होते, ऍड होत असते. म्हणजे this jumping gene does not changes its original place. हा jumping gene काही स्वतःची जागा बदलून दुसरीकडे जात नसतो. Anyway, whenever such copy of jumping gene get added at another place, another position in the genome of same cell, it bring about modification in the structure of DNA and modification, alteration in the genetic code. Now, above all these three different types of mutation are reflected in the messenger RNA and subsequently in protein synthesis. म्हणजे हे जे तीन वेगवेगळ्या प्रकारचे म्युटेशन्स आहेत त्यांचा परिणाम मेसेंजर आरएनए वर झालेला दिसतो आणि पुढे जाऊन प्रोटीन वर झालेला दिसतो. बिकॉज आफ्टर ऑल द मेसेंजर आरएनए transcribed on such mutated gene. Listen, the messenger RNA transcribed, synthesized on such mutated gene. ज्या जीनचं, ज्या डीएनएचं म्युटेशन झालेलं आहे अशा म्युटेटेड जीनचं ज्या वेळेला ट्रान्सक्रिप्शन होईल आणि त्याच्यावर मेसेंजर आरएनए तयार होईल तर that messenger RNA also shows certain abnormal or I will say additional or new nucleotide sequences and it affects the protein nature, it affects the process of protein synthesis. Let us consider example. आपण उदाहरण घेऊन समजून घेऊ. First of all, let us talk once again about the gene or point mutation. I told you that gene or point mutation means addition, deletion or replacement of only single base pair of the gene. It leads to the disorder sickle cell anemia. Sickle cell anemia. This sickle cell anemia, you know, it is the blood disorder due to the abnormal formation of hemoglobin and abnormality in the structure of RBC. We have studied. This sickle cell anemia is caused due to the gene mutation or point mutation. Means due to only alteration of single base pair. Alteration of single base pair lead to the change in amino acid glutamic acid to the valine. व्यवस्थित मी समजून सांगतो. आता मी तुम्हाला म्हटलं की जीन किंवा पॉइंट म्युटेशन म्हणजे काय. We know that gene or point mutation means it is simply addition, deletion or alteration in the single base pair. Now, whenever it occurs alteration in the single base pair, it also leads to the alteration in the single nitrogen base of messenger RNA. Alteration in the single nitrogen base in the messenger RNA. Because we know after all messenger RNA is synthesized on the DNA template. मग या ठिकाणी आपण कल्पना करू की या डायग्राम मध्ये असणारा हे जे दोन स्ट्रँड्स आहेत त्यापैकी हा जो स्ट्रँड आहे 3' to 5' स्ट्रँड it is supposed to be the template strand. हा टेम्पलेट स्ट्रँड आहे आणि या टेम्पलेट स्ट्रँड वर मेसेंजर आरएनए तयार होणार आहे. लुक एट द डायग्राम. सपोज दिस इज मेसेंजर आरएनए. नाउ the sequence of nitrogen bases in this messenger RNA synthesized on this DNA template would be according to the nitrogen base sequence on template strand such as uracil, cytosine, guanine, adenine, uracil, cytosine, guanine and adenine. Okay, because we all are known that the adenine is complementary to the thymine, but in RNA thymine is replaced by uracil. So in normal condition the messenger RNA synthesized on this DNA template should have such nitrogen base sequence. Okay, but if it occurs the change in single base pair, Means instead of this A and T, if G and C, this nitrogen base pair get introduced or replaced. याचा अर्थ नॉर्मल डीएनए मध्ये असणारी जी नायट्रोजन बेसची पेयर होती तिची जागा दुसऱ्या नायट्रोजन बेस पेयरने घेतली, that is G and C. So obviously what will happen? The uracil in this normal messenger RNA will be replaced by C, isn't it? And thus such messenger RNA, here I am going to show, such messenger RNA will show some different type of codon. Will show some different type of codon. Please look at the diagram for proper understanding. Suppose this messenger RNA is having direction 5' to 3'. Same is I am going to write here. Let me write the sequence of nitrogen bases for our understanding. We know that the first codon is always AUG, but temporarily just to consider U, C, G, then A, C, U, A, C, U, C, G, A, and so on. Now which was the initial nitrogen base here? Initial nitrogen base was T, okay, and here that T is replaced by the C. ठीक आहे, सुरुवातीचा नायट्रोजन बेस या ठिकाणी जर समजा डीएनए टेम्पलेटवर A होता आणि पर्यायाने त्याचा कॉम्प्लिमेंटरी या ठिकाणी मेसेंजर आरएनए वर U होता, पण आता मात्र त्या युरॅसिलच्या ऐवजी जागा घेतलीये सायटोसिनने. So ultimately the nature of codon in which this replacement took place also get changed. Okay, मग सुरुवातीचा नॉर्मल कोडॉन काय होता? A U U. ओके, आपण कल्पना करूया की समजा AUU हा कुठल्या तरी A अमायनो ऍसिडला स्पेसिफाय करतोय, but now due to gene mutation that AUU get modified to the ACU. Okay, ACU. So definitely it will code for some another amino acid, that is B, isn't it? The same thing happened here in sickle cell anemia. In sickle cell anemia, only due to the modification in single nitrogen base pair, the glutamic acid is replaced by valine amino acid at the 6th position of beta chain of hemoglobin, leading to the disorder sickle cell anemia. So here we have discussed about the example of gene or point mutation. Now come to the next, that is about the addition or deletion of DNA chunks at the time of crossing over, or maybe due to the jumping gene. If more than one nitrogen base pair get replaced, either added or deleted, which are not divisible by 3, which are not divisible by 3 or which are not multiple of 3, then it leads to the modification in the nature of all the codons beyond the site of mutation, and ultimately it changes the nature of entire polypeptide chain. How is that? Let me explain you. मी आता असं म्हटलं की समजा एकापेक्षा जास्त अनेक नायट्रोजन बेस पेयर्स जर डीएनए मधून निघून गेलेत किंवा ऍड झालेत किंवा रिप्लेस झालेत, मात्र त्या बदल झालेल्या नायट्रोजन बेस पेयरची संख्या तीनाच्या पटीत नसेल तर, मीन्स that nitrogen base pairs which are not either divisible by 3 or we can say not multiple of 3, then it changes all the codons बियॉन्ड द साइट ऑफ म्युटेशन, ज्या ठिकाणी म्युटेशन झालंय त्याच्या पुढच्या सगळ्या कोडॉन्स मध्ये मात्र बदल घडून येईल. कसा? लेट मी शो यू. लुक here, for our convenience I am going to draw this segment once again separately. Consider here is one gene and these are the nitrogen base pairs such as AT, GC, CG, TA in this manner and so on. Here I am going to write the complementary nitrogen bases. ठीक है? Now as usual, suppose this is 5' to 3' strand, this is 3' to 5' strand. Okay. Suppose this 3' to 5' is the template strand on which messenger RNA is synthesized. 3' to 5' हा टेम्पलेट स्ट्रँड आहे ज्यावर मेसेंजर RNA हा सिंथेसाइज होणार आहे. नाउ इन दिस टेम्पलेट स्ट्रँड, if few nitrogen base pairs get added abnormally, but their number is not divisible by 3 or the number of that nitrogen base pair is not multiple of 3. So consider, suppose here at this position, या ठिकाणी कुठेतरी या ठिकाणी 4 नायट्रोजन बेस पेअर्स ऍड झाले असं आपण कल्पना करू. G C T A C G A T. ठीक है? These 4 nitrogen base pairs get added here. Now ultimately the nucleotide sequence will modify. या ठिकाणी the nucleotide sequence जी आहे, नायट्रोजन बेस पेअर्स ची जी सिक्वेन्स आहे या जीनची ती बदलेल. ठीक आहे? आपण फक्त कल्पना करा की या ठिकाणी ही ऍड झाली. But before the addition of this nucleotide sequence, if the mRNA gets synthesized on it, then its nature was different, and after the addition of these nucleotide base pairs, the nature of messenger RNA will be different. Okay? How will be the nature of messenger RNA synthesized on such a normal DNA template? नॉर्मल DNA टेम्पलेट वर जर मेसेंजर RNA तयार झाला तर त्याचं नेचर कसं असेल इथे मी मुद्दाम बाजूला लिहितोय. कल्पना करा नॉर्मल mRNA 5' 3' end. आपण त्याची नायट्रोजन बेस सिक्वेन्स, न्यूक्लियोटाइड सिक्वेन्स ही मांडू शकतो कॉम्प्लिमेंटरी टू द टेम्पलेट स्ट्रँड. सो लाईक दिस A या ठिकाणी T आहे म्हणजे त्याला कॉम्प्लिमेंटरी असणार आहे A. देन सेकंड G C A ला कॉम्प्लिमेंटरी युरासिल देन एडेनिन देन ग्वानिन सायटोसिन युरासिल देन एडेनिन सायटोसिन ग्वानिन एडेनिन युरासिल like this. And we know the triplet of nucleotides on messenger RNA is known as codon. मग या ठिकाणी हा पहिला कोडॉन आहे असं आपण म्हणूया. सेकंड कोडॉन, थर्ड, फोर्थ अँड फिफ्थ या ठिकाणी 5 कोडॉन काढलेले. We know that each codon specifies single amino acid. But if these 4 nitrogen base pairs get added at this point, या ठिकाणी समजा ते ऍड झाले DNA मध्ये, so what will happen? The nature of template strand will modify. टेम्पलेट स्ट्रँडचं जे नेचर आहे ते मॉडिफाय होऊन जाईल. तुम्हाला या ठिकाणी कल्पना करता आली पाहिजे. मी दाखवण्याचा प्रयत्न करतो. समजा या ठिकाणी आता हे ऍड झाले CGAT. Okay? Now on this modified or altered template strand, modified or altered DNA template if mRNA is synthesized, then how would be its nature? त्याचा नेचर कसा असेल बघूया आपण या ठिकाणी लिहू. This is supposed to be the abnormal mRNA. Okay? As usual, 1, 2, 3, 4, 5, 6, 7. First 7 nitrogen bases same as A, G, C, U, U, A, G. Okay? But now here the new nitrogen bases get added in the DNA template, so obviously their complementary nitrogen bases will come in the messenger RNA. Let me show by using another chalk. Complementary to the C, G. Complementary to the G, C. Complementary to the A, uracil. Complementary to the T, A. And then beyond mutation, again the same sequence of nitrogen bases. C, U, U, A, C, G, A and U. Now let us mark the codons, nature of codons on this abnormal mRNA. आता या abnormal mRNA वर असणाऱ्या codonsचं नेचर आपण शोधण्याचा प्रयत्न करू. Look, the first codon is AGC, like normal codon. Second codon UUA, like normal mRNA. Then next one, third is GGC. Which was the third codon on normal mRNA? GCU. But now here is the GGC. Okay? Codon, nature of codon get replaced. Same is true again. The next one here on abnormal mRNA UAC, but here in normal mRNA the fourth codon was UAC. ठीक है? In normal mRNA the fifth codon is GAU, but here the fifth codon is UUA and one more codon get added, CGA, and one more is there. Means what? The entire sequence and nature of codons get modified, and accordingly there is a change in the nature of amino acid, leading to the formation of some abnormal protein or new protein which is not actually expected during the process of translation, during the process of protein synthesis. Now the new proteins formed may be harmful because they can inactivate different biological processes, even they can kill the cell also. One more thing I want to add. Sometime the sense codons may get changed to the nonsense codon due to this mutation. काही वेळेला सेन्स कोडॉन हे नॉनसेन्स कोडॉन मध्ये पण मॉडिफाय होऊ शकतात. We know nonsense codon means the codon which does not specify any amino acid and which stop the process of protein synthesis, and these are usually present at the 3' end. सर्वसाधारण नॉनसेन्स कोडॉन हे मेसेंजर आरएनएच्या 3' एंडला असतात. आणि या ठिकाणी समजा हा कुठला तरी पहिला कोडॉन आहे किंवा तिसरा कोडॉन या ठिकाणी मी दाखवलेला आहे UUA, हा सेन्स कोडॉन आहे कारण तो कुठल्या तरी अमायनो ऍसिड साठी माहिती आणतो. पण म्युटेशन झाल्यामुळे समजा त्याचं रूपांतर झालं कुठल्या तरी नॉनसेन्स कोडॉन मध्ये, म्हणजे UAC च्या ऐवजी त्या ठिकाणी समजा तयार होऊन गेला UAA. You know UAA, it is the nonsense codon. काय होईल? The process of protein synthesis is stopped at this codon. There is no further synthesis of protein. There is no further formation of amino acid chain. पुढचे त्याच्यानंतरचे जे कोडॉन्स आहे ते रीड होणारच नाही आणि पर्यायाने पुढे अमायनो ऍसिड्स जोडले जाणार नाही, प्रोटीन सिंथेसिसची प्रक्रिया पूर्णच होणार नाही. And that's why such type of mutations are also known as frame shift mutations. It means the mutations caused either due to the incorporation of jumping genes or maybe due to the recombination leads to drastic change, major change in the nucleotide sequence of DNA, and ultimately it also brings about drastic change, major change in the sequence of codon in mRNA, which is transcribed on that mutated DNA. Ultimately, the sequence of amino acid, or we can say the basic framework of the protein which is to be synthesized, get completely altered, get completely shifted, and that's why such mutations which bring about the complete change in the basic framework of sequence of amino acid in the protein which is to be synthesized are known as frame shift mutations. Now let us talk about one more little topic, that is tRNA as adapter molecule. tRNA as adapter molecule. Friends, in 11th we people have studied in very detail about the RNA and different types of RNA. We know that the RNA is one of the nucleic acids. RNA, it is single stranded and polymer of ribonucleotides. It means that structurally RNA is made up of single strand of ribonucleotides. Also we know that RNA is divided into two main types, such as the genetic RNA, that is the RNA which acts as genetic material in few viruses, and non-genetic RNA means the RNAs which play important role in the process of protein synthesis. Now non-genetic RNAs are of three types: mRNA, rRNA and tRNA, and all these three types of RNAs play very important role in the process of protein synthesis. Now here we have to discuss why is tRNA called adapter molecule. You know, tRNA means transfer RNA. T stands for transfer RNA. Why this is so? All of you are known. Because at the time of protein synthesis, at the time of translation, tRNA, it brings the different types of amino acids from the cytoplasm at the site of protein synthesis, that is near the ribosome. Transfer RNA हा सायटोप्लाझम मध्ये असणारे वेगवेगळ्या प्रकारचे अमायनो ॲसिड्सला रायबोझोम जवळ वाहून आणत असतो, ट्रान्सफर करत असतो, किंबहुना म्हणून त्याला ट्रान्सफर किंवा tRNA असं म्हणतात. According to the R.W. Hole, the tRNA appears just like trifoliate clover leaf, and he proposed the structure of tRNA. Let me explain you briefly how is the clover leaf structure of tRNA proposed by Hole. Look at the diagram. According to this clover leaf model, transfer RNA has three arms. Here I am going to show in diagram. Each arm has stem. This cylindrical part represents stem and apical loop. This globular part, spherical part, loop-like part is known as apical loop. So I said that according to the Hole, according to the clover leaf model proposed by Hole, tRNA structurally shows three arms and each arm has stem and apical loop. In the stem region, the complementary nitrogen base pairing is seen, while in the apical loop, the nitrogen bases are unpaired. Here I am going to show. In apical loop region, the nitrogen bases are unpaired. Now, out of these three arms, the middle arm is known as anticodon arm, and the loop of this middle arm is known as anticodon loop, because this anticodon loop shows presence of group of three unpaired nitrogen bases which are togetherly named as anticodon. We know that anticodon is nothing but the group of three unpaired nitrogen bases present on the anticodon loop of tRNA, and the anticodon is complementary to the codon in mRNA. In addition to that, this tRNA shows shorter 5' end and longer 3' end. The 3' end has CCA sequence at its end. It means at the 3' end there are presence of cytosine, and adenine nucleotides. Okay? This 3' end is also known as amino acid binding site. Amino acid binding site. Now, the scientists have discovered and proposed that the anticodon present on the tRNA has ability to match with the codon in mRNA. It means the tRNA can read the codons present on the messenger RNA, and accordingly this tRNA binds with specific amino acid at their 3' end. Here I am going to show. Suppose here is any particular type of amino acid. So the tRNA, it binds with the specific type of amino acid at its 3' end, and that's why this 3' end is also known as the amino acid binding site, and it carries that amino acid near the ribosome for the process of translation, for the process of, we can say, protein synthesis. And here, as the transfer RNA, it binds and carries the amino acid to the ribosome, therefore it is known as the adductor molecule. Now, why these tRNAs are necessary for the transportation of amino acid from cytoplasm up to the ribosome? प्रश्न निर्माण होतो की या ठिकाणी या ट्रान्सफर आरएनए ची का गरज आहे सायटोप्लाझम मधून अमायनो ऍसिड्सला रायबोसोम पर्यंत वाहून नेण्याची. कारण the amino acids cannot read the codons present on messenger RNA. कारण मेसेंजर आरएनए वर कोडॉन्सच्या स्वरूपात जी माहिती आलेली आहे, बेसिक इन्फॉर्मेशन आलेली आहे डीएनए कडून, ती अमायनो एसिड्स वाचू शकत नाही. That can be read, that code language can be read, which is in the form of codons, by only tRNA with the help of anticodon. Okay, and that's why the transfer RNA, it binds with the amino acid and carries that amino acid to the ribosome. Basically, the cloverleaf model proposed by Holle is two-dimensional, but in actual structure, the tRNA, it appears inverted three-dimensional L-like structure. I am going to show here, somewhat like this, somewhat like this. You know that this structure, it looks like the inverted L. This is the original structure, actual structure of RNA, it is three-dimensional, but for our convenience, the clover leaf model was proposed by the Hole. So here we have discussed one more little topic, and here ends this topic, here ends this lecture. Thank you, and all of you take great care of yourself. Bye.